All right, guys, welcome back. Lecture 19 in ZOO3649, Evolutionary Genetics. Today, we're continuing with our third lecture in the section Population Genetics. This is a basic introduction to population genetics. Remember, first lecture in population genetics, you got how to work out allele frequencies and genotype frequencies. Second lecture, you got Hardy-Weinberg, and then you had to figure out also allele frequencies and genotype frequencies. And you figured also which forces of evolution could be causing deviations from Hardy-Weinberg equilibrium and how they cause deviations from Hardy-Weinberg equilibrium, those 7 points, remember? Now, in this lecture, we're going to unpack some of those forces in a bit more detail, and we're going to see how they interact with each other. And this is great stuff. And please pay attention because all of these lectures are going to be very important when you are doing assignment number 2 and also practical 6, 7, and 8 in the lab face to face. So we're going to model selection. We're going to model different fitness of different genotypes in practical 6. In practical 7, what are we going to model? In practical 7, we're going to change the size of the population. We're going to have a big population. We're going to see how many alleles get lost through genetic drift. And then we're going to make this population smaller and smaller and smaller. What are you going to find? You're going to find that more and more and more alleles are being lost as the population size goes down. And then you see the power of genetic drift. Okay? In practical 6. And 7. Practical 8, we're going to look at how selection and drift interact with each other. Which one is more powerful in a population? Is selection, Darwin's main idea of evolution, is that the most powerful force of evolution? Or is genetic drift more powerful? The argument between selection and genetic drift in evolution is an age-old argument, and we're going to get into all of that in the next few exciting lectures in this population genetics section of 3649. It is a very good, and this 3 lectures really give you the core foundation and what you really need to understand if you want to seriously consider a career in genetics. All right, so let's see how these forces interact now. Okay, you know there are 4 forces of evolution. We've been through these. Okay, mutation, selection, drift, and gene flow. Now we want to talk about, remember we talked about Ron Fisher in the Fisher runaway hypothesis. Well, it's the same guy now, right? It's the same guy. Fisher was a very clever guy. He used maths to figure out various things about evolution. And Fisher was intrigued by Darwin's idea of selection, and he wanted to mathematically look at selection. So Fisher, and remember, he's the one who came up with the runaway hypothesis for sexual selection. So Fisher was a very, very clever guy. And here again is a picture of him, not so young, but also not so old. Fisher said that continuous variation in allele frequencies, as you see in the natural world, this variation provides the means for natural selection to work. Because the variation is what? Different things. And selection, the environment, chooses which of those things is the fittest one, right? And so that's why you have variation, which is continuous. It's a continuous variable, okay? For example, you have allele class here, or height. Remember, we had height down here in a previous example looking at this. And you can see this variation, and you can see on what pieces of the variation selection is working. So for example, here, okay, when you have selection against the extremes, okay, you have stabilizing selection, but selection, you can see where natural selection is getting rid of alleles, okay? And what will happen when natural selection does that, right? Look at this continuous variation. It is getting rid of the variation at the one end, all right? In B here. And B causes the mean to move that way. It causes the mean to move to the right. And so it causes the mean to move in a direction. So it's called, B is called directional selection. And C, what is C called? Where the continuous variation here is being affected. Selection is working hard on this one, this part of the variation, around the mean. So it's selecting against, those guys are not fit. Selection is saying, no, this variation here and this variation here, that's fit. But this variation in the middle is not fit in C. And so what happens after generation? Those guys get fewer and fewer and fewer until they are gone. And you actually have two different populations. So this is called diversifying selection. So the point Fisher was making is that variation in allele frequency is absolutely necessary for selection. That's also what Darwin said, right? But he didn't know about alleles, Darwin, right? He thought it was gametes and so on. But he was right. He said variation is necessary. And now Fisher comes with mathematics and he says, it's the variation in alleles that is necessary. Okay? So it's the different alleles that are being selected by natural and sexual selection. So the rate of change by natural selection is a function of the amount of variation in the population. Okay? Selection needs variation. Darwin said it. But now Fisher is going one more and saying, I've done it with mathematics. And you can see that the rate of change via natural selection is depending on how much variation is there to begin with. So if you have more variation, right, in alleles, then selection can cause changes faster. If you have less variation, selection can cause changes slower. This is very important, okay? And this is a product of the fact that natural selection absolutely needs variation, like a brother needs a sister. Natural selection needs variation to happen, okay? And the variation is in the different alleles. Now we know what is the actual variation. It's the alleles that a natural selection is acting on, okay? But Fisher came to the same conclusion as Darwin. If natural selection was the only force of evolution, you remember in the days of Darwin, we didn't know about drift, we didn't know about migration, we didn't know about mutation, right? We only knew about selection. So that was the first evolutionary force that Darwin came with, right? So in the days of Fisher, they were trying to put maths to this. And he figured out, but wait, the same thing that Darwin figured out, okay? But Darwin did it without maths. And Darwin said, if natural selection is only going to select for the fittest individual, then eventually we will have a population with only one allele, the fit one, only one allele. So in other words, you will have a population with no variation. And what happens to a population with no variation? Finish, okay? No more selection. Selection can't work. You're dead. If the environment changes, everyone's dead, right? So that's a tricky one, no? Eventually, all the alleles will be the fittest allele, and there'll be no variation. That's what Darwin said. Eventually, you will just have the fittest phenotype, and then you will have no selection, nothing to select. You need two at least to select. If you have one, that's it. You can't select anything. That's what you have. And if it's not good enough for the environment, all of them are going to die. Okay, so selection has a problem with variation, even though it needs it, because selection leads to no variation. If selection, unless it's selection, but we will get to balancing selection in a couple of lectures' time when we're talking about the neutral theory. Okay, balancing selection is the only selection that can increase alleles in a population. All other selection reduces alleles and gives only the fittest allele. So selection eventually will lead to no variation. And Fisher, unlike Darwin, was able to come with a solution for the question. Now, what was the question of questions? Now, as things get more progressive, the question of questions gets different, right? During Fisher's time, the question was, how can natural selection work if it will lead to no variation? Okay? And this is where the beautiful story of Sewell Wright comes in. Okay, Mr. Wright, here in picture, and you can see behind him there's some mathematical equations. Right, he was a mathematics guy. He was an American guy. He's dead now. But he is a very, very important person in evolutionary biology, because Sewell Wright was the very first guy to come with the idea of genetic drift. Nobody else thought about it. Even Fisher, who was very clever, Yeah? Even Fisher was very clever, was scratching his head, trying to say, how can we have selection when it will lead to loss of variation? Wright comes at that moment and says, Murray, selection is not the only force of evolution. We have another force called genetic drift. So, genetic drift, what is it? It's basically anything that changes the allele frequency from one generation to the next due to random chance. Something that was not predictable. Something that was simply random. Like a car accident. Two people got married, they have a car accident. They can never have kids because they're not there anymore. So their alleles, what? Is not going to the next generation. Right? So that random event of the accident has gotten rid of the alleles from the gene pool, and the next generation is not going to have the same allele frequencies. That means genetic drift has changed the allele frequency, has caused the population to evolve. Wright was the first guy to recognize that just random chance can cause evolution as well as selection. Okay? And the idea that it could be an evolutionary force, an important evolutionary force, especially when the size of the population gets small, Wright was the guy to propose that idea. And he used maths to show that the effect of genetic drift was inversely proportional to population size. What does inversely proportional mean? That means if the population is small, the effect of drift is big. Okay? If the population size is big, the effect of genetic drift is small. Okay? And I want to give you a demonstration of what, just so that you get that into your head and you will never forget it. Okay? Because you can't forget this. This is basic population genetics, and you need to understand this to get to the next sections of 3649 even. Right? So you better understand here what is genetic drift and why is it affected by population size. Let's look at this example here. All right, we got two colors of beetles. Okay, you've got green beetles and you've got brown beetles, right? You've got 3 green beetles and you've got 6 brown beetles, right? So you can actually work out the frequency of the green beetles. What is it? It's 3. Let's just work out the genotype frequency of these 3. It's 3 divided by the total, which is 9, right? So we've got 3 out of 9. Okay, that's a big proportion of the population. So now what happens if you have a random chance, an accident? Some careless person who didn't even realize there were beetles on the ground steps on 2 of them and kills them, right? Now what happens? Look, those 2 are gone. Now you've only got 1 green beetle, and you've still got the guys who didn't get squashed, the brown ones, right? But you've only got 1 green one left. So now the frequency of the green allele is what? So the frequency of the green allele here was 3 divided by 9. Now the frequency of the green allele is what? 1 divided by 7. So it is dropped remarkably. It is dropped a huge amount just because 1 accident happened to just 2 individuals, and the allele frequency goes boom down. Why? Because that population is small. Now let's consider the population was not 9. Let's say the population was 1,000. No? And they were, instead of 3 beetles, there were 300 beetles, green. And there were then what, 300, 1,000, we had 700 brown beetles. So now we have the same. We have the same proportion, same allele frequency, right? But the population is huge. Now what happens when some careless person is not watching where he's going and squashes 2 green beetles? Yes, the frequency will change. The allele frequency will change of the green. Why? Because you've gotten rid of 2 of them. But remember, In the small population, there was only 3 and you got rid of 2. In the big population, there's 300 and you got rid of 2. What change in allele frequency will there be? Yes, there will be a change, but the change will be tiny because you only took out 2. Now, instead of 300, you got 298, right? So 300 divided by, what is the total? 1000. It will give you 0.3. What about 298 divided by 1000? It will give you pretty much the same, 0.3. So that's the point, right? In a big population, you can have a random event. It's not that important. It changes the frequency that much. Whereas if you got a small population and you lose 2, whoa, that frequency goes like this. Huh? So you see how genetic drift, it is more effective, or the effect of it is much stronger in a small population compared to a large population. Okay, so we need to understand drift. Okay, we need to understand that it is a central concept of biology. Okay, just like selection, it is a central concept. And when your population gets small, like all our endangered species, rhinos and everything that we are killing and poaching, genetic drift is the biggest factor that is affecting those populations of endangered species. Okay, you guys are studying about conservation and so on with Swanepoel and all these other guys. Well, genetic drift is something that is very, very important in conservation, because when you're conserving, why? They've already come to a small size. They've already been killed everywhere. They're only in a small game reserve. You have a few rhinos left. Genetic drift is going to be a huge factor, much more than selection in that population. We are going to have fun with this during the practicals. All right, so this shows here basically that, you know, you have the white allele and the black allele, right? And you see the frequency of the black allele is 0.25 in generation 1. And then what happens? Not everybody mates. Only the effective breeders, the effective population, actually mates. Okay? So we were going to get this NE. N is population size and E is effective population size. We're going to get to that. We're going to unpack all that in the next lectures. Okay? And the effective size is the number of breeders, because only the breeders are going to pass their genes to the next generation, right? So the effective population size is actually a way to measure genetic drift, right? The same way fitness is a way to measure selection, effective population size, the number of breeders in the population, is the way to measure drift. Because if that number is small, drift is going to hammer that population. It's going to reduce genetic diversity there. Drift never increases diversity. It can only reduce diversity. Okay? So, and here we go. Here is a graph with 3 lines, 2 red lines, one here and one here, and one blue line in the middle. So, this is exactly what your simulations in practical 7 are going to look like. You are going to start your population. Here's the starting point here, generation. Here the generations is time. You're starting at 0 and then you're going 10 generations, 20 and so on. Your time is going forward in this direction. You're watching my mouse, guys. Time is going forward in this direction. And this is the frequency of the big B allele, right? So what does big B do? Big B makes the mice black, okay? Small b makes the mice white. So when you have all the population, where, for example, here, what is here? 0 is here, right? And what is that 0? It's what is on the y-axis? The frequency of B, big B. So when the frequency of big B is 0, what is the frequency of small b? It's 1, right? Because 1 minus any allele frequency gives you the other allele frequency, because they have to add up to 1, right? So here, at this point, big B allele, which makes the black color, is 0. No one animal has big B, right? Which means everybody is white. And on the other hand, here, when big B is 1, every single rat or mouse will be black. because they got the big B color gene, right? And what will small b be at this place where big B is 1? Small b will be 0. There will be 0 white alleles, right? So now let's go back and start our simulation. We're all going to start our simulations. We start our simulation, and we're going to say the allele frequency starting at 0.5, right in the middle. 0.5, right in the middle between 1 and 0. So we started off here, and we say our population size is 10. Okay. Let's see what happens to the allele frequency of B after 50 generations. What happens? Let's take one of these red lines. In one case, it goes up, it goes up, it goes up, it goes up. Oh, suddenly comes down, goes up, suddenly comes down, up, down, up, and eventually goes all the way up. Okay? You see how this curve has got sharp changes. Yeah? That's because the population size is small. N is equal to 10 here. Okay? So the population size is so small. That's why you get sharp changes. Genetic drift has a big effect. Same here with this red curve. Look what happens with this one. It goes down, it suddenly goes up, it goes down, it suddenly goes up, and then it goes down, and and then finally it goes down to 0 and stays. Of course it stays. Once it gets to 0, it stays at 0 for this population here. So what has happened here? Genetic drift has made the population go down. So that's why it's going up and down, and it's quite likely to go to 1 or 0 under genetic drift. Okay? In other words, you started with 2 alleles. Here, 0.5. If B is 0.5, what is small b? Is also 0.5. That means there are 2 alleles in the population at equal frequency. And what does genetic drift do? It pushes one allele out. On this red line, it pushed out the small b allele. And on this red line, it pushed out which one? It pushed out the big B allele. Genetic drift always wants to push out an allele. And the smaller the size, the more able the population is to push out an allele. Now we increase the population. You're going to be doing this in population by yourselves on your own computers. You're going to be doing this in Practical 7. And you're going to be writing an assignment about this, don't forget. That's why you have to watch these lectures carefully. Now what happens here with this blue line? What is different about this blue line? You can see immediately it's not going up and down sharply like those red lines, right? You see that immediately. It's more smoother. And the changes, they're not going up high. There are changes, but they're small changes. They're tiny little changes, right? And what happens? The blue line, unlike the red, it doesn't go up or down. It actually remains more or less in the middle, right? And why is that? Because the size of this blue population is 1,000. So a random chance that happens to 1 or 2 individuals, it's only going to be a small change in the frequency of the allele. It's not like I told you about those beetles that got squashed. If you had 300 and 2 got squashed, it's not a big deal. But if you only have 3 and 2 get squashed, it's a big deal, right? That's what this graph is saying. If you've only got 10, the population is going to be hammered by genetic drift until only 1 allele remains. If you've got a population of 10, the population is going to be hammered by genetic drift until only 1 population or 1 allele remains. You see with these 2 red graphs. But if your population is big, you can have a lot of drift is not having an effect. Why? It's having an effect, but it's not a big effect. It's still, after 50 generations, you still have both big B and small b in that population because the size is 1,000. When the size is 10, you will never get a situation where you can go for more than 50 generations and you still have both alleles. Genetic drift will get rid of one of them. Okay, and that is why genetic drift is so powerful. Look how powerful it is here in changing the allele frequency when the population size is small. So genetic drift becomes a very, very important evolutionary force in changing allele frequencies when the population becomes small. When the population is big, drift is not a problem. Okay. So this is in answer. This is in answer to the question of questions of the time. Okay, if selection is only getting rid of the alleles, if genetic drift is getting rid of alleles, selection is also getting rid of alleles, but keeps the fittest one. Genetic drift doesn't care which allele it gets. It can throw out the fit one, it can throw out the unfit one. It doesn't care. It's random, right? But selection is only selecting one allele, right? So selection will always lead to a reduced variation, and genetic drift will also lead to reduced variation. So how can they interact so that selection does not always give you at the end only one allele, so that even with selection you can still have variation in the population? Remember, this was a big problem for Darwin, and it was a big problem for Fisher, because both of them said, well, selection of the fittest allele will reduce the variation, right? Yeah. And now Sewell Wright comes and says, but with genetic drift, even though genetic drift also reduces variation, it's not the same variation, it's random. So if selection and drift can interact with each other, they can actually manage to keep variation in the population. Let's have a look at how that. Genetic drift solves Fisher's problem, right? And which was also Darwin's problem. The problem is selection is reducing the genetic variation to the fittest allele. Drift can prevent this by changing the frequency of the alleles at random. Okay? Right, I figured this out and introduced this idea called the adaptive landscape. Look at this beautiful graph. It's in 3D, okay? And you see a landscape. You see mountain. You see a mountain here. I see a little mountain here. And then you see deep valleys. Another and a very deep hole here. You see that? So this is actually called a fitness landscape, okay? When fitness is on this y-axis or the z-axis, and the other two axes are the allele frequencies of two different genes, and we see how these two genes form this landscape. So at certain frequencies of these two genes, you can have a high fitness. However, at different frequencies of the very same two genes, you can have very low fitness, right? And this is this fitness landscape that Wright introduced to biology, this adaptive landscape. So basically, any change in the frequency of gene A or gene B, you will move along this landscape wherever that frequency is. Here, the frequency is here and here on this axis. So wherever the frequencies are, where the frequencies change here or here, your position on this landscape will change. You could be at the top at high fitness. If the frequency changes, whoop, you could be at the bottom at a low fitness, right? And that just depends what the environment is doing. So how can allele, right, who is on a fitness peak, like say this one here, but it wants to achieve higher fitness to go up here, but it can't, because in order to do that, it has to go down, it has to become less fit. And as soon as you become less fit, you're out. So as soon as you come down from the mountain, you can't go back up, because your fitness will take you out of the population, because you're getting low fitness and whoop, you're out of the population, right? So you can't go from one peak to another very easily if there's only selection. Now, if there's only fitness, now this is where genetic drift comes into its own. Sewell Wright said, and here you have that graph, that genetic landscape. It's the same as this one here. Just we are graphing fitness versus one allele frequency, not two. So it's not a 3D, it's a 2D. So we got fitness here and allele frequency there. So we don't have the third frequency. So here you see it's a peak. There's a fitness peak here and there's a fitness peak here. Okay? So the butterfly has a long proboscis for putting its tongue down, that proboscis down the flower and eating nectar, right? So this population here with 3 centimeter long proboscis is stuck on this fitness peak, right? Because what's happening? The environment is changing, right? So the nectar, the flowers are getting longer because the environment is changing. The flowers are getting longer. Now this 3 centimeter tongue proboscis of the butterfly is not long enough to go all the way down, right? So how can that butterfly evolve a longer proboscis and get a higher fitness. So basically, how can it go from this fitness peak here to that fitness peak there? See this fitness peak? The butterfly has a long, much longer proboscis, and it can reach the nectar at the very base of that flower. It can put its tongue all the way down, right? But how does the population go from here to here without going all the way down and coming up? Remember, when you go all the way down, what happens? Your fitness becomes lower and lower and lower. And so actually, when you go down and you have a low fitness, the chance of you going to the next generation becomes very low. So as soon as you go down off this peak here, there's nothing stopping you from disappearing. So you can't come back up to get a longer, to the other fitness peak there. You can't come up. Just with a world with just natural selection, it's not possible to do this. It's not possible to go from here to there. It's not, watch my mouse. It's not possible to go from here to there, right? Because you have to go to low fitness, and when you go to low fitness, you're dead. So you're never going to be able to get up here, right? So how does selection work if you can't do that? It's the same as saying, as soon as the variation is reduced to the fittest allele, there's nothing to select, no more evolution, selection, no selection, dead, right? That's the same problem as this problem. How do you go from here? You've got one allele in the population. Now you need variation to select, so for something to select so that you can evolve something more, right? So you can't go down this fitness trough. You'll die. You can't come up. You're finished. You're extinct here. So what do you need? You need genetic drift, okay? If a population is stuck there, right? If there's only selection, there is no way to go down, yeah? But genetic drift, because genetic drift, remember, it doesn't select the fittest allele. Even if selection is saying, hi, Connor, this allele is going to the next generation, drift comes and says, oh, no, population is small, this one is going to die. Even if it is the most fittest. Genetic drift doesn't care about your fitness, right? So when drift does not care about your fitness, it changes the allele frequency at random. So as soon as you have drift and selection together, you are able to get, just by chance, alleles which are not the fittest ones in the population, remaining in the population, because genetic drift does not get rid of them. Okay, it gets rid of fitter alleles maybe, and genetic drift randomly keeps those alleles, and so it allows you to move from one peak to the next without going all the way down and becoming extinct. Okay, you see that? That is why selection cannot be the only, Darwin's selection, natural selection, sexual selection cannot be the only evolutionary force. Okay, it's only one evolutionary force. Everybody says, oh, Darwin, evolution. But he only came up with one force. All these other guys came up with the other forces, okay? And that's what you are learning about here in 3649, okay? An allele can drift towards a higher frequency once it is high enough. Selection can drift to a higher frequency, and selection can take over and push it up to the next fitness peak. That is why you cannot have a world where only natural selection is working. That world doesn't work. Evolution can't work in that situation. It needs drift. Do you see now why it needs drift? If you are asked a question in an exam, why does selection, what would happen in a world where there was only selection, and what, how would I phrase this? And what other forces could help maintain genetic variation in populations? This would be how you would answer that question. Okay, so we come to gene flow. Okay, we're not done with the drift. You've done with my description of what is genetic drift, what is selection, how they interact, very briefly. Now I'm going to also briefly talk about migration, gene flow. Okay, migration is the fourth evolutionary force, and it's a simple one, right? But it's the only evolutionary force that needs two populations. You need a population where you are leaving, you're a migrant, right? You have to leave a population to migrate. Then you go to another population. And so you have the other population, which is receiving. Okay? So it's the donating population, the one that where you leave, and then there's the receiving population, right? So it can also change allele frequencies. But it's unlike the other two because it needs two populations, right? Donating and receiving. So you see here, we have a population. Let's call this population of balls a population, and each allele, there are 4 colors. Each ball has a different color, and they say there are different alleles, okay? So then they're equal numbers. You see this pie here? It says equal pies of white, blue, green, and orange. That means all, if you count them, they're same. They will be the same number of every color ball, basically. That is what this is saying, right? Because they're exactly the same numbers in each ball. So now how, and so that's the frequency of the alleles. It's 0.25, 0.25, 0.25, 0.25, right? So what about when you have gene flow? You can have emigration, so leaving, okay? So let's just say these 2 orange here, they've left. And you can also have immigration. That means arriving. So let's just say these 2 green have arrived. Now, in this generation, what are the allele frequencies? They are not the same as those ones. Look here. You have decreased the frequency of orange from here. You see how big it is here? It's smaller. Why? Because you've taken away 2 orange. They've left the population. And what about green? Green has become much bigger. Green was small here, it's become bigger. Why? Because 2 individuals have arrived and they had the green allele, and so the amount of the frequency of green is much bigger, the frequency of orange is much lower, and the frequency of blue and white, they have not changed. But what has gene flow done between this generation and this generation? It has changed the allele frequencies of the population. That is why we call it a force of evolution. Okay, so now how does migration interact with selection? Yeah, what happens when you have selection and you have migration at the same time? Remember that selection is trying to find the fittest allele, right? Migration, someone migrating in might not have the fittest allele. Okay, so despite selection, you can have weak alleles coming into the population. So there are some general rules. If there is no selection or the selection is weak, right, then migration from one population to another can lead to the both allele frequencies becoming the same. Okay? Please, guys, take this into your heads. This is important because the 2 populations, they're different. But if you have gene flow between them and selection is very weak, because what normally happens, you have gene flow and then selection says, that allele does not fit, and it nails that allele, right? That's when you have strong selection. But let's say we don't have strong selection. We have 2 populations, gene flow between the 2 populations, but there's no strong selection. So if you bring an allele, it's not really selected out. If you bring a allele there, it's not selected. This is a weak selection or no selection, right? So what happens? After many generations of migration, both populations look identical to each other, right? They've become homogenized. They've become the same because there's so many people going back and forth. All right? It's like, so for example, if we have our population in the city and the population in the rural areas, there's a lot of migration between those populations. So the allele frequencies should be very similar because there's so much migration between rural areas and urban areas. Okay? So, but what if there is selection, right? So, if migration is bringing in the same alleles that selection is favoring, okay? So if migration brings individuals with fit alleles, then these alleles will be much higher frequency in the receiving population. And now we're talking about the first one was when there's no selection or weak selection. The second one here, and the third one will be if there is a selection, right? So if the second one is now if migration and selection are both playing for the same team, right? When they are both increasing the fittest allele. When a migrant comes, they have the fittest allele, and selection is selecting that allele. Then that allele will become higher frequency in the receiving population. But let us say that migration brings in an allele that is not the fittest allele. And selection is like, that's going down. That one is not fit. We're not sending that one to the next. It's not going to survive the struggle, right? So we will get rid of that one. So if migration brings an allele that is not fit, selection can nail that. And so what will happen? Migration can bring it in, but selection takes it out. And so what happens to the allele frequency? Nothing. Okay? The allele frequency doesn't change. So depending on what is selection doing and what alleles are coming in through migration will determine whether the population allele frequency changes or whether the population frequency is not affected at all. Okay, so we'll leave it there, and in the next lecture we'll talk more about the interesting concepts of population genetics.